Hey there, my name is Richard. Today I'll be talking about amyloid-beta peptides and its role in the pathophysiology of Alzheimer's disease. This is my physiological systems presentation and I hope you enjoy it. What is the normal physiology of amyloid-beta peptides? Amyloid-beta peptides are an amino acid residue cleaved from much larger amyloid precursor protein, APP, by the actions of alpha secretase. Recent literature has suggested that amyloid-beta protein is a normal part of the innate immune system. Researchers from the Massachusetts General Hospital recently published a study that expression of human amyloid beta predicted mice, roundworms, and cultured human brains against lethal infections. This is in lieu with a growing perception that amyloid beta has many of the qualities of an antimicrobial peptide, AMP, a small innate immune system protein that defends against a wide range of pathogens. AMPs fight infection through several mechanisms. One fundamental process involves forming oligomers that bind to microbial surfaces, which then clump together into aggregates, preventing the pathogens from attaching to host cells, and allows AMPs to kill microbes by disrupting their cellular membranes. Recent evidence have also indicated that amyloid beta may play a role in controlling synaptic activity. Induced stimulation of hippocampal neurons in brain slices increased production of amyloid beta, providing negative feedback by depressing the synaptic activity. Without such a depression, synaptic activity would become so excessive that it would lead to excitotoxicity, and thus apoptosis, which is the death of the cell. Other less-known potential activities of amyloid beta also include activation of kinase enzymes, regulation of cholesterol transport, protection against oxidative stress, as well as being a transcription factor. So although the activities of amyloid beta are numerous and yet to be fully elucidated, but what happens when there's an overexpression of amyloid beta peptides, or conversely, its impaired removal in neurons? Well, they aggregate and become proteotoxic to the cell, forming hard insoluble plaques, as shown in the image to the left. Beta amyloids can bind strongly to a receptor on nerve cells, setting in motion an intracellular process that erodes their synapses with other nerve cells. Furthermore, this compromises the integrity of normal cellular mitochondrial, endoplasmic, or cytosolic metabolism, leading to in this diagram to the left, you can see the normal cleavage of APP by alpha secretase. However, to the right of this, you see the abnormal cleavage of APP by beta secretase or gamma secretase as a result of mutations in APP or presenilin 1 and 2. This leads to excessive amyloid accumulation and thus toxic oligomeric aggregations. There are several theories regarding the mechanism of action leading to Alzheimer's. One of them is known as the amyloid cascade hypothesis. The amyloid hypothesis was formulated through several observations in past studies. It proposes that mutations of APP, presenilin 1 and 2, and/or inherited familial Alzheimer's disease genes leads to increased amyloid beta production, as shown in the previous diagram. In particular, toxic oligomeric amyloid beta 42 and amyloid beta 40 peptides, which are the most common isoforms of the peptide, initiate the neurodegenerative cascade, as you can see in the diagram to the left. Increased aggregation consequently leads to increased amyloid beta plaques, as well as tau phosphorylation and neurofibrillary tangles, NFTs. This causes decreased synaptic activity and neuronal cell death, as reflected by gradual cognitive decline, the most commonly observable pathological hallmark of Alzheimer's and dementia in general. A recent study published in September last year showed that an antibody, aducanumab, resulted in a significant decrease in amyloid beta plaques in both mouse and human trials. As a result, cognitive deficits were rescued in patients over a 1-year period. It is a human monoclonal antibody that selectively targets aggregated amyloid betas. In a transgenic mice model of Alzheimer's, aducanumab is shown to enter the brain, bind to parenchymal amyloid, and reduce soluble and insoluble amyloid betas in a dose-dependent manner. Aducanumab cleared plaques of all sizes, suggesting that aducanumab triggered clearance of pre-existing amyloid-beta plaques and prevented formation of new ones. Several mechanisms may be involved in aducanumab's amyloid-beta lowering activity. The clearance of amyloid-beta deposits was accompanied by enhanced recruitment of microglia. Thus, phagocytosis played an important role in A-beta clearance in these models. Activated microglia appeared to encapsulate the remaining central dense core of plaques in treated animals and humans, possibly isolating them from the surrounding neuropil. It is commonly thought that soluble amyloid-beta oligomers rather than monomers or plaques may be the primary toxic species. And considering that amyloid-beta plaques might be a source of amyloid-beta oligomers, this suggests that treatment with aducanumab might slow their release into neuropil, thereby eliminating the toxic effects on neurons. This suggested mechanism of action is consistent with results gained from the clinical trials, which have thus far been largely successful. Now you might be wondering why these results are so important. Alzheimer's affects more than 100,000 Australians alone. Recent and past drug failures have highlighted the complex nature of this disease, and treatments targeting novel pathophysiological aspects of Alzheimer's is required in hopes of discovering a cure, or at the minimum, slow the effects of neurodegeneration. These results are important in that aducanumab could be the newest approved drug suitable for human use since more than 10 years ago. Hope you enjoyed the presentation, and thank you.